First, I want to thank David for making what is very complex science more or less understandable. And if I can more or less understand it, that's like the low bar. Because there's a reason I went to law school and not to study this sort of stuff. So I'd like to go back to one part of sort of the history of all this that David did not cover. And that is the fact that, we talked about the period around the end of the 80s into the early 90s and the focus on fetal tissue transplantation. And don't need to repeat what David has said about the results of that research, which were at best ineffectual. But there was an earlier chapter about 20 years before that where reports started to drift out from various places that experimentation research was being done on live human fetuses, many of whom were destined for abortion. This became a big issue after Roe versus Wade, which occurred in early 1973. And almost immediately, and actually this effort was led in part by the late Senator Edward Kennedy of Massachusetts, to stop this type of research. Amendments were put forward in 1973 to ban research on the fetus outside the uterus. The National Research Act, passed in 1974, established a national commission for the protection of human subjects in biomedical and behavioral research, and also prohibited research on a fetus from elective abortion until that commission, which had been established by the law, had reported back to Congress. A panel convened in 1971, so some overlapping things here, had reported that research resulting in no harm to the fetus would be permitted so long as the research might benefit other fetuses. But the restriction that was passed in 1994, the statutory restriction, took precedence, and all such research was banned. So I referred to a statutory standard before. Basically, it's a standard of minimal risk, and also a standard that requires that any, even in order for any such research to be justified, it has to be shown that the knowledge is sought to be gained by the research cannot be achieved by other means. So this early effort more or less froze this type of research, I would say, fortunately, and so it's not something of concern, but it of course overlaps with the push, as David described it, to go beyond the 14-day rule. Obviously, research within the 14-day rule is still experimentation on a living human entity, human being, albeit embryonic. And any push to go beyond the 14-day rule takes us further into this territory. So even though there are statutory restrictions on this type of research in the context of abortion, living fetuses, et cetera, there is a push by some researchers to essentially do the same thing, but justify it on the grounds, well, these are very early human beings, if they even would consider them to be human beings. So I guess the basic point here is that Congress addressed this issue almost 50 years ago, 45 years ago at least, and now we're sort of back, and this is coming through the back door through advances. I put that in at least partial scare quotes, that would that will allow experimentation beyond the 14-day rule. So let's go, since we talked just now about vaccines, I think it's time, getting towards the end here, to discuss what has been a contemporary issue of controversy within the past year or so, actually going back almost to the beginning of the effort to launch Operation Warp Speed in the development of vaccines. I think some of the earlier statements on this go back as far as March of 2020. So it's as soon as this pandemic started and the need for a vaccine became apparent, there were statements put out that that the vaccine effort ought to be ethically driven, that we should not be relying on the cell lines originally derived from an induced abortion. So this controversy kicked around. Again, some people see this as an advantage, some people see it as a disadvantage. We'll discussion for another time of having a magisterial authority to help settle some of these issues. But the Vatican Congregation for the Doctrine of the Faith in December of 2020 issued a statement on the issue of vaccines and cooperation. Now, to give a little bit of background, it's very hard. Some people may have the impression that the Vatican is just ever ready to issue various thunderbolts or statements of something that's good or bad. That's actually not how it works. It takes forever. Time stops when you cross the Tiber. I was told by one person who knows a great deal about such things that getting the Vatican to issue a statement like this as rapidly as it did was another example of Operation Warp Speed. In this case, warp factor 7, I think this person said. So, it took a pandemic and all of this discussion to light a fire and get this statement out. It's rather brief. I think I have a slide that includes. Well, I don't actually, but it's a rather brief statement and can be found rapidly. But just a few paragraphs, and the summation of it is, as the slide says, that it's preferable that vaccines not be produced from the descendant cell lines, but it is morally acceptable to receive those that are so produced. Now, one thing the statement does not get into is the distinction between the messenger RNA produced vaccines and the other vaccines that are produced using the cell lines. The mRNA ones, as David said, can involve testing. The cell lines involved in testing, but not in production. So I thought it was unfortunate that was not emphasized, that there are vaccines out there that don't, that at least in terms of production are not tainted or possibly tainted. We're going to get into that issue in a moment. So here's the money quote. The kind of cooperation and evil, and this goes back, I talked a little bit earlier when I was talking about the appropriation theory, that there's another way of putting it that fits it into the more classical format of material versus formal cooperation. And that is the category of passive material cooperation, in which there's nothing that's done. There's nothing that's done by the cooperator to contribute in any way to the act of abortion, but it's passive. It's something that's received, and I think that fits in with the concept of appropriation. It's something we receive or benefit from. So that I'm not sure that that notion of passive material cooperation is coextensive with what Kathleen Caveney and others have put forward as the theory of appropriation, but at least there's some overlap there. So the kind of cooperation and evil, Vatican is saying this, is passive material cooperation. So this kind of cooperation in the procured abortion from which these cell lines originate is, on the part of those making use of the resulting vaccines, that is, those of us who receive them, remote. And both of those italicized phrases are, that's original emphasis. Um, so the Vatican is saying this is remote. So the moral duty to avoid such passive material cooperation is not obligatory if there is a grave danger, such as the otherwise uncontainable spread of a serious pathological agent, such as the SARS-CoV-2 virus. The Vatican statement goes on to say that no one is obliged to accept the vaccine, but those who refuse the vaccine have a duty to the common good to protect themselves and others from further spread of the virus. Finally, that governments and pharmaceutical companies should aim to produce ethically irreproachable vaccines. Also, governments have a duty to make such vaccines available to poorer nations. So this is where, again, I think at the time the statement was issued, the Pfizer, if not both the Pfizer and Moderna vaccines, had been approved for emergency use by the FDA. And it might have been helpful if there had been further clarification on the point of production versus testing. And I think this is something David can certainly chime in on. He's far more an expert than I am. But when we talk about the testing, I think it's important to keep in context what that means. That kind of testing is used in all sorts of human manufacturing endeavors, and I think the next slide, well, not the next, yeah, the next slide, I think. Let's see, I'm not sure it says it specifically. Yeah, anymore. Yeah. Well, somewhere there, the statement, this is a short summary of a statement. This is put out in 2021, a couple of months ago. Some of you may have seen this. Ryan Anderson, who is now the head of the Ethics and Public Policy Center in Washington, D.C., he had been at Princeton, has written a lot on gender transition issues. I think many of you may be familiar with the book, which Amazon has now taken off its list, When Harry Became Sally, which talks about gender trans, which addresses the whole issue of genders. So he's a very serious scholar on these issues, and he and others, an eminent group of people, including some folks who've lectured at CBHD, Father Nicanor from Providence College, for example, Dominican. Father Kevin Flannery was on this. Carter Sneed from Notre Dame, and many and several others with great expertise on this issue issued this statement. And this goes a little bit beyond the Vatican statement, I think, in speaking on the cooperation issue. So it's this, again, I'm just taking out a money paragraph. It's not a long statement. Again, very, very accessible, can be read in a few minutes. Common to all pro-life witnesses, recognition that the apparent elective abortion that led to the derivation of the HEK 293 cell line was morally impermissible and involved the unjust taking of human life. But the HEK 293 line currently used in scientific research, and those like it, as David has emphasized, do not contain the remains of any human being, and so its use does not show disrespect for human remains any more than the contemporary use of products such as roads or train lines that were constructed by unjustly enslaved human beings, or use of land unjustly taken shows disrespect for those victims of the distant past. So this gets to, I think the analysis here could be seen as an intersection of the classic formal material, Lagorian construct of formal and material cooperation, the theory of appropriation, and what I would consider the Flannery Thomistic approach, looking at the proximity and the circumstances of the receipt of these vaccines, and probably to some extent the production of the vaccines. I think where the statement makes a very significant point, and this can be, there might be some point of disagreement on it, but they're essentially saying that there's a moral equivalence between those vaccines such as J&J that are produced using the cell lines and the Moderna and Pfizer vaccines, which do not, at least in production, since in neither case are fetal parts used or abortion promoted. Elsewhere in the statement refers to the HEK 293 line. Again, David can talk about this. It's used for testing in all sorts of products. And again, I'm going to hope David can chime in and tell us a little bit more about that. So this leaves another question on the table. Well, let's say our analysis, whether we're using the classic Ligurian standards, whether we're using the appropriation standard, or whether we're using the Thomistic Flannery, where we focus on circumstances and proximity to evil. Let's say all the vaccines pass under all those things, but there's some people who simply just say, I have a problem with this because, at least with regard to the J&J and some other of the vaccines, that the production of these vaccines relied on the HEK 293 cell line. What's going on there? Is it just a feeling, emotion? Is it something we need to get over, or is it something more important that needs to be respected? A very interesting discussion on the website Public Discourse took place fairly recently, I think in March of this year, between Richard Stith, a now emeritus professor of law at Valparaiso University, and Melissa Michela, who is a professor at Catholic University, a colleague, maybe not in the same department, but a colleague at Catholic of David Prentice. So there's been some back and forth on this prior to this discussion, and I've just summarized some of the key points that each makes. So Richard Stith starts out with that moral repugnance reflects our sense of feeling contaminated by any causal connection to evil. This should not be ignored, or those who raise it be dismissed. Professor Michela comes back and says, this is important. Moral repugnance has to be acknowledged. It's not a sufficient guide to human action, to the morality of human action. Our innate feelings of repugnance need to be educated and examined so they may be directed to their proper object, in this case, the past evil of abortion and the use of fetal tissue without proper consent. That's where our repugnance should be directed, to what happened in the past. But those evils to which we direct properly our repugnance are not perpetuated by the use of the HEK 293 cell line in either the production or the testing of the vaccines. Richard Stith acknowledges these are good points, but he emphasizes also that the moral scruples of those who feel repugnance towards the use of these vaccines should not be overcome simply by a strong self-interest or a societal interest. The education that Professor Michela calls for will not assuage our repugnance at the evil of abortion. And finally, we should aim to avoid harm, not merely to avoid culpability. So that's reducing what they've said here. It's certainly worth a read. Again, it's not a very long read. It's on the pages of Public Discourse, so it's freely available website. If you just plug in the name Stith and Michela, you'll find it. I think this raises Richard Stith's comments, and I think he gets into this a little bit, is part of the issue. Even if we agree, and I've just put my cards on the table, pretty strong supporter of the statement that I'll be going back a slide here to the U.S. pro-life Catholic scholars. I mean, I know some of these people, and I think they have pretty much hit the nail on the head here. While they put an addendum in saying, great respect for the people who still having a problem with this, and that's and ensuring that they're not saying that people should feel morally obliged if they have a conscientious objection to nevertheless take the vaccine. They do emphasize that, and I think that's important. But I think what's also going on here is the societal pressure that says that these kinds of concerns are really without merit. And this sort of gets to all of the, I don't know if politicization is quite captures what has happened during this entire COVID period, now going on pretty much going on 18 months. We can't seem to put it behind us. But some of the ethical questions, the broader ethical questions that have arisen in terms of our entire response to this, are subject to a lot of influences that aren't necessarily very studied, aren't necessarily in keeping with the type of moral analysis that I think the attendees and certainly the presenters here would engage in. And that by that, I'm referring to the role of mass media, the role of so-called experts, whether they be self-appointed or appointed by others, kind of to tell us what to do. And I think part of the resistance, the good faith resistance to, I'm not talking about sort of radical anti-vaccination, which goes beyond the COVID circumstance. Good faith questioning about this is to some extent a reaction to that dynamic, that we're sort of told what to do and that our moral scruples about something, or our moral scruples saying, well, just to give an example, where some people have said, I have a moral scruple about some of these lockdown things because I think it's damaged. For example, underprivileged children have been denied of an education, not necessarily based on, the best of evidence. I have a moral problem with that, and sort of told by some authority somewhere, well, you sort of, that's not important. Well, I think that's part of what's going on here too. And I think it takes a certain amount of courage. While I don't agree personally with the people who feel that, this level of their moral repugnance it's not a reason I would adopt for not receiving the vaccine. I've been fully vaccinated. But I kind of understand where they're coming from. And I think that has to be, while good information has to get out there, and resistance, if any, to the vaccine should not be based on improper information. I think David has done a fantastic job clarifying those issues scientifically. I think from an ethical perspective, we have to be very careful about letting the ethical decision-making be driven unduly by the media. So here's a copy, by the way, of the page on the Stith-Moschella dialogue. So I think we're now at Q&As. I hope I didn't go on too long. I think we're right at 3 o'clock, so I think we're pretty much at the end. And if there's any questions on any of this, please. I think we on time and under budget, right?
Great. Yeah, thank you both. Great information from Ed and from David. And so at this point, please feel free to take advantage of both of their knowledge on the subject of fetal tissue research and regulations. Feel free to send a question through the chat, or you can let me know either through the chat, or if you want to signal by doing the green check mark next to your name under participants, and I will call on you if you'd like to ask a question orally. Otherwise, I'm fine reading your questions. Or if you need a clarification on a point too. I'll ask a question I received on an email that's for David, and it had to do with one of those charts that you put up from the Charlotte Lozier Institute. Someone emailed me from Hungary, from the country of Hungary, and she wanted to know, what about the Chinese vaccines, Sinopharm and I believe they're Sinopharm and Sinovac. Do we know how those are, if they use cell lines in production and their confirmation studies?
And we've got those in the list, and I can't recall. See if I can just bring it up here. But we did look at that. They're actually, I think, Sinopharm and 2 different Sinovac. And let's bring that up. As I recall, those are whole virus vaccines.
I thought there were 2.
Yeah, so Sinovac is an inactivated virus, goes under the name Coronavac. Monkey cells is how they're growing. They grow the coronavirus in monkey cells. So monkey cells for development, monkey cells for the production. They did use an HEK 293 cell test or 2, just to see if it was making the right protein. And pretty sure Sinopharm is the same. No, as far as we could tell, and how we're evaluating this is we look for the published scientific papers. Usually they will publish something after their preclinical studies, before they've even gone to the clinical trials. And so you can look in their materials and methods and look through their papers, published papers, and find out. But it's not from news stories, it's not from patents, which are not necessarily relevant to this, but on the actual publications for these vaccines. We're finding that the Sinopharm did not use HEK 293 in any of their tests, and they used Vero monkey cells for production. Just a comment, a point. Ed was talking about testing versus production and so on. We were kind of surprised at the whole testing thing. As I mentioned, there used to be just one way to make a viral vaccine. So the question was always, what cell type did you use? Simple, straightforward, that was it. But then with these newer molecular ways, and I'll pick on the mRNA ones because they've simply been in the lead, they're the quickest to make. You don't use any cells, period. It's all chemical and enzymatic synthesis in the test tube. But then we looked at their paper, and there was mention of HEK 293 cells. Well, what had happened was they designed their vaccine, and you can follow a clear timeline, in fact, as to, it was very quick. As soon as the genomic sequence appeared on the net, they could download it, they could figure out what the spike protein gene sequence was. On computer, they made a couple of changes in that sequence to make the protein more stable, because they're not putting it on a virus, it's just the protein by itself. Started production within a week of designing that sequence. But then once they had their vaccine made and before they went to clinical trials, did it make the protein? Did it make the protein they want? Did it, if we put it into animals and vaccinated animals, did the animals make antibodies, neutralizing antibodies against the protein? And it turns out one of the tests is what's called a pseudovirus test. Instead of making a lot of infectious virus in my lab to do these tests, I make a pseudovirus. It's a faux virus, but it has that spike protein on it. And then I just test sera for antibody titer from the animals. A lot of people made pseudovirus in HEK 293, including all of those warp speed candidates that we looked at. So it was kind of surprising to see, but as Ed mentioned, HEK 293 is used in a lot of tests. Now, I do think I should say some people have gone to a ridiculous extent when they talk about its use to test everything. I've seen some lists where they, for example, put aspirin in. Well, let's be realistic here. Aspirin was first synthesized in the lab in 1897.
Yeah.
AGK-293 was first grown in 1973. Now, if somebody decided, hey, I want to do a science fair experiment and see what effect aspirin has on AGK-293, if they did that test in 1985 or just last month, I don't really think that has an ethical taint on aspirin. So, but they are used in a lot of tests. Mostly where you're going to see them are in production of what are termed biologicals, making therapeutic proteins, making some genetic therapeutics, or making, you know, viruses and these sorts of virus vectors. So,
Are there any consumer products, David, generally available tested using this?
Yeah, I'd have to go back and check that to be sure. When you say consumer products, are you thinking of like,
I don't know,
Pepsi?
cosmetics or even food or you know, just testing maybe for food safety or anything? I've just seen brief
Yeah,
references. I really haven't looked
a
into it.
number of years ago, there was a group who genetically added a human taste receptor to HEK293 cells so that if it caused a certain reaction in the cells, it would cause some sort of change in the cell medium. And they were using that kind of taste testing. Pepsi was the one that floated up. There were several companies, and it was a company called Cinemix.
Yeah.
You know, that's another kind of testing. It did rely on the cells, not that it needed to use HEK293. And it's, again, I think a point that we need to look at, that there are a lot of things going on that we don't know about.
Right.
But there are also a lot of alternatives.
Yeah.
I also want to throw into the mix that there are a lot of scientists that have no clue about the ethics of the origin of HEK293. They use those cells because it's what they were taught with as a grad student, or the convenience. The guy down the hall has it in his freezer, or he's growing it. And somebody said, you know, or I'm trying to do an experiment just like these other guys did, so I can replicate and see if I, you know, so it's, but there are a lot of folks. I have a good friend, strong pro-lifer, who did an experiment with some cells he had ordered, and then to his chagrin realized one of them was an abortion-derived cell line. Yeah. And of course, he never used it again, and even put a thing in his paper about it. But you know, there's a lot of education that needs to take place, and then there's a lot of, I call it discernment, that people need to use in terms of, is there really an ethical taint? Is there really an ethical connection here? Or am I just getting, you know, carried away? And, you know, we don't try and push people one way or the other. We say, you know, we want you to make sure you have the most recent and accurate information, but that's a decision of conscience for people to make themselves, related to themselves, their family, and their community.
Yeah.
I have a question from the audience. So Ed, did you want to add something to that?
No.
Okay. Relatedly, what is likely to happen with regard to the vaccines that are made using WI38 cells and MRC5 lines? Once those lines are deceased, will they use the HEK cells to continue producing the vaccines, or will they try something else?
It'll be something else. HEK is really not well set up for the kinds of viral vaccines that MRC5 and WI38 use. And there are some people who have said, oh, we need something just like it. And in fact, in 2015, the Chinese started a cell line called Wolvax2. Basically, they thought it might be a replacement for a WI38. but I haven't seen that used for, frankly, anything, and certainly not for any vaccines. Nobody's even attempted to do that in the US. The more likely outcome will be switching to a molecular technique for those types of vaccines, so you don't use any cells, or switching to a licitly derived cell line. And there's a great example out there now. Sanofi used to make their polio virus vaccine in, I think it was MRC5, but one of these old abortion-derived cell lines. About 2 years ago, they switched, and they use that Vero monkey cell line now. So I think as time goes on, you're going to see a migration away from those older cell lines, and we'll still, though, be faced with, what about these, like the HEK293, the adenoviral producers, the biologics producers. There are alternatives there, and people use those alternatives already, but it's kind of ingrained in a lot of scientific labs. And so, we've been trying to encourage several labs to develop a better cell line. It can't be just the same, because people won't switch.
Right.
They're going to look at it pragmatically. It has to be a better cell line, a more efficient cell line to do what they want it to do.
Yeah. Build back better, right?
Yeah. To coin a phrase.
To coin a phrase.
All right. If people have any questions that come to mind about anything throughout the day, feel free to send me in the chat. While they are thinking about their questions, I had one, a point of clarification, and it was something, David, that you mentioned briefly, but I know people have questions about this because I've received those questions too. You mentioned, so there's something ethically wrong with human-animal chimeras, but transgenic mice, or mice that have like a human immune system, doesn't cross that species boundary. So this isn't the mice that, like, say, for example, have the ACE2, the ACE2 gene does not cross that species boundary with the same kinds of concerns as, a pig with a human brain or something like that. And I was just wondering if you could clarify why those are two different, why those are morally distinct.
And I think what most people will do is they'll point back to what entails species identity. And especially if we think about human beings. What if I look at you, or you do something, how do I recognize you as being human? And it's not just DNA per se, because we share a whole lot of genes with other species. But so you start to look at very human specific traits. Now, DNA does factor in if it's in terms of reproduction. If you can reproduce another human, okay, yeah, that you're probably a human. But now if you've got a pig that makes human sperm and another pig that makes human eggs, they're obviously not human, but they might potentially reproduce a human. It's interesting, just as an aside, that the answer to that by the folks who really want to make a lot of chimeras is, we'll just keep them from reproducing. We'll keep them in separate cages. Won't be a problem. I helped design our AAA LAC facility, animal facility, when I was at Indiana State. And the best, finest way you design these things, there's always some mouse or rat that escapes, that figures out how to get out, wanders around and so on. There's no 100% certain way. So cognition then, of course, another trait we would think of as humans. And in point of fact, there are some human mouse chimeras they did a number of years ago where they could actually, even with what many would consider a fairly minor contribution of human brain cells, the mice did better on some of their little mouse tests. Yeah, so we're not talking about cartoon mice here. We're talking about beings that somehow are acquiring different capabilities, almost human capabilities. And then the other thing being just recognizable characteristics. And so I used the example of, you know, the monkey with human hands, human looking hands, or the sheep with a human face, and so on. Yeah, those obviously, besides being just creepy, yep, do blend, you know, the species identity. If I look at you and I can't really tell what species you are, that's a problem.
All right, I don't, let me check my email and check, and it looks like I don't have any additional questions from anyone here. So with that, I will wrap us up. Thanks, David and Ed. CBHD is blessed to have you guys on our advisory board, and we are blessed to have you guys teach on these topics again for us. Also, big thanks to Brian for being our host, our Zoom host, tech guy, and coordinating everything and all the stuff that he does behind the scenes. In August, we will be publishing a special report on fetal tissue research that covers much of what is being covered today. So Ed and I are the editors on this. We're partnering with the Charlotte Lozier Institute. So you'll see articles by some of them, including David Prentice.
Well, thank you very much. And I hope we'll see all of you at our conference this evening, beginning at 7 o'clock Central Time. And if you have any questions between now and then, just please let me know.
I guess all I have to say in closing is, I should have said it earlier, but I just regret that we weren't able to do this in person, and I look forward, if CBHD will have me back, to doing these things in person. I come cheap, so I think that's the main attraction here. So, and
that's all you had to say.
Yeah, communicate, exactly. Communication is not connection. I think we've communicated, hopefully we've communicated well today, and but it's not the connection that we have attending in person. So for those of you who should come to these things in the future, look forward to seeing you in person. Thank you.
Amen.
Wonderful. Well, if that's everything, students, you're free to log off. If presenters would stick around for just a moment, a couple items just to cover with you guys that, since I have you in person, would probably make it easier.
Okay, very good. Thanks.